Okay, back again to that solution. So for the regular secondary structure prediction, it made the segments predicted much longer, much more realistic. The same is true for the transmembrane prediction. Now for the transmembrane prediction, what happened is that helices got twice as long as observed. What do you believe I did as the developer? So I'm not a very clever person, right? I'm just a hacker. What would you do? So you see, this is half the length you want. You do this, is twice the length you want. What do you do? Oh, 3 times the length you want? No, that I didn't do. That's an interesting idea. Oh, you could try to look for the correlations. I tried that later. But this actually didn't work. And there's a reason for that.
Try to oversample short helices.
Oh, that is, in some sense, that is a more advanced version of that idea, I would argue. I did not try that. It's a clever idea. It's what I'm telling you. I don't have this. I'm not as advanced. But I can maneuver you into the same situation. It's just that I tell you on the day that you have to hand in your thesis, there is something wrong with it. Now what do you do overnight? In this particular case, really, it was something that happened overnight. What do you do? It's twice as long as you want. Yeah, you know the answer, right? Just say it.
Yeah, you just define it as half the length of the output.
So that's an interesting one. No, that is in some senses. That is more clever, in some sense more clever and more radical. So I simply cut it, okay? On average it's twice as long as I want, so I said if it exceeds a certain length, cut. Dumb, huh? But it actually worked very well. And it stood there as the best method for many years, until I found something that was also simple and improved over it. And that essentially is the same idea that we had here, getting through two different predictions. So when you look at the two output units, transmembrane, not transmembrane. What I show here is residue, the running sequence. And for every single residue, I have a probability of predicting that it is non-transmembrane. It's high, that goes down. This one goes up. They are mirror images, one minus the other one, right? So now you can take this one here, the transmembrane unit, and you can perceive it as like an energy or a probability for forming transmembrane helices. Now what could you do? Again, here you see an example like that. When you go over the 0.5 value, this helix here is way too long. So what I did not tell you is how long the protein is. I believe this is 300 residues long. So you immediately see that this is a very, very, very long transmembrane helix. Any idea how long transmembrane helices are? Ballpark figure.
25 residues.
Perfect, perfect. In fact, this morning I saw an abstract where somebody had analyzed all transmembrane helices, saying that the average is 25. My response was, this cannot be. So I don't believe it, but it's you're right on with the latest manuscript separative on this matter. Okay, so what could you do?
Increase the threshold.
Yeah, that you could do, but so this is what you have, and now you have to get the prediction of helices. How many helices do you have in there? How do you see that?
Yeah, 3 peaks.
This, in fact, that's the way you do it. So this one is not a peak. So you filter out everything that is more than half or something. And what do you call this one here? There's a peak.
It's more like a...
So you do something like that too. So you do a sort of high pass filtering kind of thing. Okay, But if you think, this is a very valid approach, and you could indeed do that. What I would challenge you is you get into the same sort of fitting business that I said cut half. This is, you begin to do the same, you use more intelligence for so if you do a hack, have it really simple. If you use intelligence for a hack, be careful. Maybe you can do something more with your intelligence. I would argue then. But this is the kind of thing that we typically all tend to do. So we find a quick solution, we see we need a correction on the quick solution, we do a little bit of a correction, and half a year later we have a correction of correction of correction. But I see your point. That would be an alternative way of doing it. But there's something more generic in this. And the more generic situation that you can somehow see, let me just say it in a way that is very, very simple to perceive. I could just find the optimal path through that landscape compatible with the idea that helices have to have a certain length. So maybe the average is 25, they have to have a minimal length, and then they have a maximal length. What's the reason for a minimal length of a transmembrane helix? Why can it not just be 4? Why does it have to be something much higher than 4? Yeah?
It has to go through some membrane.
It has to go through the membrane. It has to span the entire membrane, right? At least transmembrane helices. Those that go through have to go through. It's sort of a little bit circular. And then at some point, in fact now we get into the other issue, if they are very, very, very long, actually they get back into the membrane. There's an experiment from Gunnar van Heijnen in which he can show that if he makes it long enough, it is going to be reinserted as another transmembrane helix. If you're longer than, depending a little bit on what is in between, but even if you keep going on a helix, on a transmembrane helix, if you go beyond 40, it is reinserted. Okay, as simple as that. Now, back to that, the way to do the algorithm is very simply, you label all possible membrane helices, so you put them all in there, and those happen to be all, and I label them by the average propensity for forming helix. So now, if I predict that my protein has one transmembrane helix, my best bet is this, 0.95, because that has the highest average. And again, so my segments are such that they are at least the minimal helix and at most the maximal helix that I have observed in my data set. And between these two extreme numbers, I just pick them all. Okay? Given, so if my assumption is that there's one membrane helix, it's this. Now if there are two membrane helices, now we get into the next issue. Well, the second highest in my example chosen is this. That cannot be true because I already have a membrane helix here. So I have to stay out of the membrane helix, I have to have some space in between, and then the second highest is this one. So if I believe that my protein has two, then it's these two that are most compatible with my prediction, and so forth and so on. And I simply stop when my overall probability goes down, which in fact happens to be your threshold of 0.5 ultimately. Not necessarily, because you have an overall probability for the entire landscape, so it could be well before that. But it may just be the 0.5. Algorithm is clear, right? In some sense you could argue it's a hack, but it is a very, very simple algorithm that fits the idea, find the optimal path through that landscape, given simple constraints. So you do not have that many assumptions in here. And it's very, very easy to compute. It's essentially a dynamic programming on the level of transmembrane helices, of which we have just a bunch, just several. So it's super fast. Now remember how we can assign topology? By simply the Gunnar van Heijnen inside-out, positive inside rule, meaning that there are more positively charged residues at the inside of a membrane. So once we have this assignment here, we can simply count the positive charges outside and inside, do a difference, and we get the topology. That's what the method gets. And in this particular case, So the T is very correct. What you may not remember from last Thursday, when I used the globular secondary structure prediction method, this is all an E. So in terms of globular secondary structure, I completely incorrectly predicted it. I did not see that as a helix, but I saw it as a beta strand, because it's overall very hydrophobic. But this specialist does very well. Now, what other method could you imagine to use? Not a neural network. So in principle, what I have here, if you want to look at it like that, I have some constraints. I have some rules. My system, I'm not only looking at this prediction, the initial per-residue prediction, but I'm fitting it into something because I have background rules, length of helices. I could not do the same thing for globular secondary structure because there are no constraints. Globular helices have length from 4 to over 150. In the ATP synthase, we have two coil helices that are over 150 residues long. And in fact, they form a coil, or again from Francis Crick. Francis Crick miscalled this a coil. So you have two helices winding around each other, and they are called coil. So you have this one, and then you have the ATP synthase, and this actually drills a hole in the ATP synthase. It's like a simple, so do you guys know these simple drills for kids, where you have two wires around each other, and then you have something that sits on top, and you slide it up and down, and the drill turns? This is exactly what it is. There's something sitting on top that moves up and down and drills a hole into the membrane. So these are very, very, very long helices. Another example for long helices are on tops of cell membranes. You have, again, two very, very long coils that just meet, and then they intertwine and bring the cells close to each other. Those are examples of extremely long helices, and in this case they are over 150 residues long. So for globular proteins that doesn't work. For membrane proteins it does work, but this brings us back to my question. Can you think about machine learning type or statistical or blah 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 kind of method that uses grammar?
Decision tree.
Decision tree. How would you do that?
You get a half question, like for example the length of this second helices and stuff, the average hydrophobicity and all this stuff.
Yes, but that you can put into the, what is it that you gain from the decision tree that you don't have in your network?
Simple. Very simple.
Oh, that's true. It's a simpler solution, and you can do many things with the decision tree that, in particular, simple problems are simpler to address. Certainly true. But you cannot put grammar onto it except for the tree level, the tree map. So if you know that this is the first part of the decision, are you membrane protein or not? Something like that. But this is not the type of problem that we have here. What we have here is certain length, right? And this is not, at least I don't immediately see, yes, you can map that onto a decision tree, but it's not generic. Anything else you can think about? And was that the kind, whenever you smile, Jade, then you want to say with your smile that it's totally trivial, or?
No, that is redundant.
It's redundant?
Plus.
For what? For the signal peptide?
Oh, at least you asked exactly the same question.
Yeah, for the signal peptide, I forgot. Okay. So from now on, Jade smiles, I know I said that before. So the funny thing is, I don't remember what I said yesterday. How come you read? Anyway, so the story goes back to Anna Schroch. The team was with Eric Sonheimer, Gunnar van Heijnen. And simply the point really is, in a hidden Markov model you can put in a grammar, so you can somehow see that there's something to model the globular environment. There's something else in terms of the environmental situation to model the caps. There's something, the situation in between in the membrane layer, and there's something that is loops versus non-loops, and so you have different elements that you can classify, and then you can say, well, I have to go into the helix loop, so continuous state helix, for a certain amount of time, and then I can get out with a certain probability. So you can simply tweak the probability. That means you can make before shorter than 15 you don't allow, or you allow with a very, very low probability, and then you allow to break out, and then above 25 or whatever the number is, you have a very high probability of breaking out finally. So this is ideal for this kind of problem, right? And that's TMHMM is what that was called. Here's another solution from the Hungarian, from István Simon and Gabor Tusnadi, shortly thereafter. Is in detail is slightly different, but it uses the same logic, it uses the same advantage. Now, I'm not entirely sure, I have not presented this, and I'm not entirely sure why I'm presenting this suddenly so quickly here. Typically, why do I do this? So when we assessed prediction accuracy, we claimed, I claimed in a paper published 20 years ago that these kinds of methods reach percentage of almost 90% of getting the helix right, and 86% of getting the topology of the protein right. And this was a publication that is highly cited and is totally wrong, absolutely wrong. And it took a long time for me to publish that this is wrong, because many people had published the same thing. And they all were against me publishing that what we published was wrong. I initially had only seen that what I published was wrong, but it implied also that others were wrong, and they did not like that so much. The reason, however, why this was wrong is really ultimately related to the kind of data that we all used. In order to do this, when I started, we had essentially one membrane protein in terms of structure. One transmembrane helices, the photoreaction center. There are many versions of the photoreaction center, but essentially the way you look at it, it had many chains, but essentially it's one protein. So you could not use your assessment here only on the X-ray crystal structure, but you needed low resolution labeling. And the low resolution labeling, turns out, was as good or as bad as the prediction. Okay? And that is another reason that made it complicated here to publish this. But we know today that all methods have been overestimated, and there's no single best method. There are some good candidates, and I get into these now in more detail. And that is work from Jonas Reip, which ultimately came from the question which method to use. What we know is that many methods use hydrophobicity scales. There are many advanced methods that use machine learning, HMMs and the like. There are consensus methods that simply average of all of those. Essentially, how would you choose? You would take all the data that you have. Now, comparing all the methods is not trivial. The first problem is some of these methods are very old. How would you address that problem? You would exclude some, but how would you decide what to exclude? Well, one way of deciding that is you make everything run that runs, and everything that you cannot make run doesn't run. How do you rule out that exactly the ones that are really good are the ones that you left out? Any idea? Any suggestion?
Maybe you could search for literature and which says this method is really good in comparison to the others?
Yes. So you cannot, you're a little bit in a loop here. Because if you could completely trust the literature, you wouldn't have to do the whole thing. So, but you can at least see whether there's any tendency of people who have done that before you. And if that tendency says the best method out there is exactly the one you leave out, then the best thing you can do is talk to the authors. Try to reach the authors. Okay, now the other thing here that comes when you compare a bunch of prediction methods, you have to always compare them on the same data set. And maybe some methods fail for some proteins. And the more methods you have, the more likely some of these will fail for some. Today we have many, many more membrane protein structures than before, but we still only have in the ballpark of 160. So if every method fails for 10, and you compare 100 methods, well, you do the math, right? Odds are pretty high that very little is left over. But you can do compare comparisons. One way of doing that is in the typical way that you do in biology. You put more focus on things that are recently published, which in fact tends to be also the ones that are more likely to run on your operating system, more recent. The ones that are being used, which again you would somehow get out of the literature. They're possibly the ones that are interesting in terms of the method, that are somehow unusual. Because you also want to estimate or gauge how particular ideas help to improve performance. You have to always remember that you have to compare it to random backgrounds. So what's the random background here? How do you pick a random prediction? Let's call it the motivation comes in. No?
Very trivial would be like to say 1/3 is faulty.
Whatever, so you pick the distribution that you have in your data set and you pick that certain number with a random number generator. But the problem with that is you know that helices have a certain length, right? So you will not, I mean, sort of isolate pick of a single residue as being in T is not the one to do, right? Because you do know that you have a problem there somehow and you optimize your method in some sense, and somehow the random should optimize that in some way too. So the simplest way is you say that the length distribution of your random picks should mirror the length distribution of membrane helices. That is, so yes, you can just randomly pick, but sort of randomly pick with the length distribution that you observe is still random. There's still no prediction method in there. It's still pretty dumb, right? But there's another step up from that. What is that?
Maybe using a Markov chain.
But now you're getting too advanced in your stupidity. If you call the Markov chain stupid, then that's close enough to the Markov model where you put logic in, right? It is true by the length distribution. I sort of do something like that, and what you do is just a little bit of the next step. So I take your point, but I would argue this is already a method. But there's still something that many may argue is already a method, but that I would argue is almost somehow random, because
like you have the composition of all the three structures, right? And you have composition, like the distribution of length of all the structures, and basically just assigned randomly everything.
Well, that is, so for secondary structure prediction, I call that the simplest prediction method. But again, so this is exactly what I'm sort of looking for. So composition, and ultimately what I meant is the simplest aspect of composition for membrane proteins is hydrophobicity, right? So at least you should look for hydrophobic, so pick a hydrophobic region, do some average distribution, pick your helices. That's another way. You may not call that quite random, but clearly prediction, if you do a prediction method and you spend 6 months on it, and it should at least beat what you can write down in half an hour, right? And your Markov chain is a little bit so one way of putting that, maybe that exactly is the definition. You can program it less than 1 hour over the course of a master thesis. That still is random or genial, and it's not easy to distinguish one from the other. Okay, now one issue here is the question of how many of the residues are correctly predicted. Another is the segment. Do I get the helix? How would you do that? How would you capture this? So per-residue could simply be how many of the residues are correctly predicted in the helix or not helix, transmembrane or not transmembrane. The problem with that is that most residues in membrane proteins are not membrane, so this will be highly biased by the part that you care less about somehow. But again, the more interesting one is this: how could I possibly measure that? But I'm still in the random prediction. I'm actually saying what I said before. This is essentially, what I just mentioned, put it to words and call it semi-random. Now, this is no longer what we do. We use only 3D structures. So if we used only structures in the PDB, would that completely solve my problem? Could we take every single structure in the PDB? So there, maybe you, even when you do look at PDB, you want to look at resolution. So maybe you don't want to take the ones that have a very low resolution, and there are many in there that do. If you have something that says 6 angstrom, maybe you don't want to take it. Maybe since we are talking membrane proteins, 2.5 is going to make you happy, where otherwise you would say more higher resolution. But so there is some payoff. This is just an example of a structure that has issues. Let's call it like that. The other issue, when you look at the PDB, how do you get the membrane helices out of there? Do you know that? I briefly mentioned it.
N+3 stopping like basically there's this.
Yeah, you have a helix. You have a DSSP helix. Is the DSSP helix mapping to? Can you just look at DSSP to see the helices in a membrane protein? No, you cannot, because it could be globular helices. How do you distinguish in a protein between the membrane helix and the globular helix? You look at the lipid in the 3D structure, right? The problem is the lipid is not in there. You don't see the lipids. So you cannot do that. How do you know where the lipid sits? And actually the answer is a guessing. So you see sort of a hydrophobic cuff. You see where the sequence has a sort of hydrophobic window. When you look at the structure, you sort of see it. And you can make machines learn that, what you see. Not through machine learning, but essentially you trace what people call a hydrophobic cuff. So the end of helices that are enriched in hydrophobic residues on both ends, and that assigns secondary structure. But this is in fact not that trivial. You cannot directly read it off the 3D structure. And the other thing, of course, you have to reduce redundancy. Here's an example. Do you all feel that you see the membrane helix? That you see, and clearly you somehow see it up here, right? In fact, in this particular case here, there's a helix that is aligned with a lipid. But in principle, all of you see this clearly, right? But even here you sort of see it, right? So visually okay, and in fact you can write algorithms that mimic this. Now, there are 2 databases, OPM and PDB-TM. They essentially do what I said. They differ. So what do you do with the difference? One way is you say, O is nicer than P, so you take that one. One way is you take the later one. One way is you take the earlier one. Whatever preference. What else could you do? Well, this is what we did. And this, I'd argue, essentially, so the problem is, so it began with the argument that we could not really say that either of those two is clearly better. We saw strength in both of them, visually comparing some of the structures. And then we decided this is the solution. What else could you do?
Just take the overlap.
Exactly. Look only at the predictions where they both agree. The reason why we didn't do that is because it made the data set smaller. Plus, we argued So in some cases, one was better than the other, where they disagreed. And maybe prediction methods picked that up. We have some evidence for it, we have some evidence against it. I still don't quite know the answer to that question. But anyway, this is what we did ultimately because of this, the size. We reduced the resolution, so we took only high resolution structures. Per-residue is sort of trivial. You just measure how often the particular residue is correctly predicted, and this one here is for helices. So we essentially have unit T here, two-state accuracy is percentage of observed and is percentage of predicted, which essentially means how many of the helical residues that you have observed do you picked, and how many of the ones that you pick are correctly predicted. Okay, so record, precision, accuracy, coverage, there are different words for that. Different methods. Those are the two different values. You see error bars. Gray are different levels of random. That is hydrophobicity. This is where we sort of pick the point according to something like hydrophobicity, but here we use only hydrophobicity. So what do you observe? And the different colors are different methods. Can I have some observations?
All methods are better than random.
This is very true. So the dumb random, all methods are better. When we get to this sort of more intelligent random, sort of hydrophobicity, this statement, but this is really hydrophobicity, so it's really not random. Absolutely right, Hauke. But the next observation would be not all methods are, in fact, here's a method that is not better by this scale, at least than the simple hydrophobicity. Next observation.
The probabilities are really parallel in a way that you can basically just use the heterogeneity with already almost 80%, right?
So you say that they all are very similar, is that what you said?
The improvement using fancy methods are really Pareto in the sense that you can just use
They are really what?
Not Pareto.
So it's not impressive what you gain by putting a lot of time into it. Is that a translation of what you said?
Yeah.
Okay.
Some methods like feel worse.
This is clearly what I took away from this. So many of these methods are PhD projects. Many of these are 4 or 5 years projects. So you can't 1, 2, 3 times 4, 5. There's a big amount of work that goes into it. And this is done. This is okay. This is an hour, maybe 2 hours programming. But it's on this scale. It's surprisingly little that we do over it. I'm not entirely sure whether I see anything else. Maybe there's an outlier here. Maybe that would be observed. Maybe there is an outlier here. Maybe here. But this is per residue. So let's look at it per segment. Let's look at it on the level of the helix. Okay, now so in some sense we have predictions like this that don't make sense. So we have to somehow find a way to look at per segment predictions. And let's just look at it here. So this is the transmembrane helix observed. This is the transmembrane predicted. In this particular case, it's very simple. 100% right. The example is chosen like that. But what if I predicted it until here? So how would you, average transmembrane helix, as Hauke says, is 25. So what if that would be the prediction? Would I get that helix right? So don't look at the slide, just at your intuition. So you already, or let me use a different color so the black is the observation. Oh well, it's blue, but and the red one is the prediction. Okay, everybody's happy with that prediction. What about this one? We don't know.
Partially correct.
So now we introduce 2 states. A segment is right, wrong, or partially correct. We could do that. So we could essentially, and this, the most extreme way of that would be, so I will look at the overlap. So I call this one N observed, and this is N common NC. So I do NC by N observed, right? The common by the observed. So I essentially measure the fraction of the observed helix I get, and I sum over this fraction for every single observed helix. That is one way of doing it. Now, the problem with that way of doing it, say this is my prediction. Now what do you do?
I would argue that the predictions in the middle of the helix should be more accurate than at the end.
Ooh, that's this.
Should maybe put some kind of distribution above the helix, like a Gaussian distribution. If the ones in the middle are higher weighted than the ones on the outside, and then you can just, I don't know, multiply it or...
Smarty pants. Nobody ever did that. This is absolutely compatible with what we observe. When we have differences, they are exactly at the ends, and your Gaussian or whatever distribution you have would fit to that. In fact, this is exactly what Gunnar von Heinja, I don't know, I briefly mentioned this method, TM-sec, that he published in '94, that essentially uses this reality. But nobody ever used that to assess the prediction accuracy. Great idea. Nobody ever did it. I don't know how that works. But it's a great idea. But in this particular case, your point again is I wouldn't do that, I would do my Gaussian. Fair. But somebody else. What would you do with this one? And don't say I would do the Michael thing. So how do you treat this one? In the Hauke language. So in the Hauke language of the fraction. So would that count to the fraction? So would this be, let's call this NC right and NC left. Would I do this? The problem with that one, if you said yes to that, then the problem with that one is somebody predicting a membrane helix like that is totally unusable and still would give a relatively high, the way I construct it, say is 50%. While a method that has 50% overlap, arguably not in the center where it's most important, but still has a membrane helix that is just 10 residues removed, I believe this is much more useful than that. And in that way of counting, this would not be reflected. So what you could do is you could argue, okay, I count my helix only once. So I map predicted and observed helices, and I have one map, whatever is the largest overlap. Okay? This is in fact one way of doing it. And we have been doing this. What in the past people have done, and they had reasons for that, is they argued I match the helix correctly if, again, helices are in my universe about 20 residues long, or 20 to 25, let's call it that way. So if they have 3 residue overlap, I call it correct. And I count every single helix only once. Okay? This is the way historically has been done. In this particular case here, we sort of sharpened this or made this a little bit more conservative by arguing that the overlap has, so that they can differ by at most 5 residue on either sides, on either side. Okay? So you're allowed to be 5 residues off, and then we count it right. In some sense, implicitly, it handles somehow your idea, because you allow deviations at the ends, and you thereby implicitly force the center to be most likely to be aligned. But it's not the implementation of your idea. Anyway, that's the way it is. And now, this is what I get. So the QOK now is the fraction of proteins for which all helices are right. Okay? And it's the relatively small data set of proteins. That's why the error is relatively high. And what you observed before is still valid. They are all clearly better than random. Not so much better than hydrophobicity alone. And now we see that some methods sort of stick out or do better. But this is on 95 proteins. Many of these methods have been developed on those proteins. That's a problem, because over the years everybody used whatever they published, used whatever they had available. So in fact, some of these methods had more proteins available than others. Yeah, more recent methods and older methods, right? How can I sort of get that out of the equation? How can I sort of find something that is sort of equally fair to all of them? This is a very simple answer. If you have a simple answer in your head, just give it. It may be the right one. I'm sorry. That was yeah, whatever.
You retrain the method that is fine.
No, that you cannot do. So the definition of that you cannot do is you want to do something that you can do. This typically is not a master thesis. This is, so assessing methods is more type of a bachelor thesis. That means you have less than 3 months. And in reality, you all have done your bachelor thesis. You understand that less than 3 months means actually tomorrow. You just started and it's already the end of it, right? And reprogramming 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 methods, no way. Okay?
Tested a set which was available, which was also available at the time where the oldest method was developed.
Yes, that is a great solution. That's PhD 1. I mean, these two are the two oldest methods in there. So one is from me. So no. Yeah.
Maybe use a new data set that wasn't at all.
Yes. The problem with that one is, so this is the old data set, and I don't know why the number of proteins is higher here. There used to be 90, and now it's 146, I can't remember why. There must be some explanation for that. Actually, they also go more apart. I don't know why. Anyway, but this is new. Problem is, now we have 44. So the set is dropping. Problem is now the error. So now we really see differences in the methods, but the error goes up so much that it gets difficult to make clear points. But the first thing that you observe is that everybody drops. Conclusion? What's the conclusion from that?
You're overfitting.
Yes. Until you remember that there's a method in there that used only 1. 146 is still larger than 1. So it never had them to overfit.
Does it actually make sense that the random and semi-random actually decreases as well, because they can assume the distribution, right?
That's another way in which you sort of could. Right.
I mean, there is.
What does that indicate? Yes.
Would you repeat it?
What?
I just didn't understand.
So he, okay, so whoever didn't understand, can you please tell me what else you see in here that is strange? Don't repeat it. Luckily he was mumbling, so not everybody here heard it. There's something else here that you could stumble over. He just stumbled over it. But everybody else who didn't hear it can stumble again. Anybody? So I told you these two here, red and orange, have not changed. Have also changed by almost, so a little bit less than some of the methods here, but they also dropped, and they have been using only one one protein, so they cannot have been. So let me go a step back, Michael. What you said is what you immediately suspect, and 99% of the cases the correct answer. It's just in this particular case it was not. And you can see that because these could not have been optimized. But there is another, even if I had not told you that these two methods had only used one, there's something else that is sort of fishy, or that could have given it away, that something else must go on. You heard it, or you know it?
I hear it.
Yeah, so you don't qualify. Who did not hear what? Oh, nobody didn't hear it. Okay. It's unfair. Here, that drops too, right? The random. Or the semi-random. Hydrophobicity drops too. So there is in fact something that is going on here. It's a tougher set. So the newer proteins are more difficult. Why could that be the case? I have not said that. I have mentioned some of those issues, but I have not clearly said that. So something was easier in the old set. Why do you believe? What? Any suspicion?
Maybe they were shorter.
They, in fact, so shorter is not quite the right thing. But these here, I take from your argument the word length. These, in fact, I said that membrane helices typically are 17 to 25 residues long. Those 146 proteins fit that profile much better than those. There are shorter ones in here and longer ones. Overall, these here are typically helices that are orthogonal to the membrane. Some of those go at angles. We had membrane protein structure determination is difficult. People began to do that better and better and better. And the better they got, the extra crystallographers, the more we saw things that were not standard, that were more complicated than we had thought. So there are things out there that are more complicated, that are not, in fact, hydrophobic and are membrane. And there are things that are clearly long-running hydrophobic that are not membrane, in both directions. And that makes the task more complicated. So this is one of the reasons here. There is a difference between kingdoms, so eukaryotes and prokaryotes. And most likely the explanation is, this is because more proteins that I trained on are eukaryotes than prokaryotes, right? Makes sense to you? Does it? It shouldn't. It's total nonsense. Because any kind of determination of anything about a protein that is eukaryotic is more complicated than for prokaryotes. That's why most human proteins are first studied in bacteria, or for the homologs, not the human protein. It's much, much simpler. Any assay you do. Also assays that lead to structures. At least 10 times easier. So it's rather the opposite. We have more of these than of these. So why this is the case, we actually don't know overall. Now we get into one of those things. This is a figure we published, and I, as one of the co-authors, have the right to call that nonsense, but I just don't know how to do it better. And you tell me now why it's nonsense, okay? So what I show is the performance for all proteins that have more than 5 transmembrane helices, 2 to 5, and 1. Again, what I plot is the fraction of proteins for which all transmembrane helices are correctly predicted. What's nonsense here?
To compare them in one plot, because it's probably to get the whole protein right is for one transmembrane helix easier than for 5.
Exactly. Now comes the problem. So you want to correct for that, right? So you somehow normalize. Unfortunately, there are different ways of normalization. And they give very different answers how this plot looks. So whenever you have different choices and they give very different answers, you're in the typical situation where you don't know what to do. So you present the raw data and you say, don't use it. And anybody who in the future, so actually for years and years and years we didn't publish this, and then people publish it again and again. So at least now we can say this is nonsense, here it is. So don't publish in the next paper that single membrane proteins are predicted better. We still put the number out, and we did not know how to correct. Yes? What?
Why do we measure it on the level of proteins and not on the level of segments, that you say, okay...
But it's on the level of segments. Ah! That's why. So what you see here is for all the methods, the fraction of all of those for which we get the number right. So no mistake. And then 1 over, 2 over, 1 under, 2 under. So most methods get mostly the number of membrane helices right. They may not get the place, they get the right position, at least within plus minus 5. But the number of helices they get right. Now the interesting bit is exactly where they get it wrong. And so, but on this plot that you implicitly somehow suggested, because on this plot you don't see anything, that's the answer to your question. So the story gets more complicated, and I actually in this particular case don't have a solution. But here's something else. Can we, is the question that is answered here, identify that a protein is membrane helical protein, as opposed to a globular protein that doesn't have membrane helices? And what you see here is for eukaryotes, the degree of accuracy for a bunch of methods. Impressive, right? Very impressive. Anything wrong with that? So the numbers are right. So in this particular case, I'm not going to give you the red mark, say it's all a lie, real data is published. But it's only one side of the coin. What is the missing side of the coin? We developed methods that predict membrane helices. The methods were trained on membrane proteins. In membrane proteins, they find membrane helices. The real question is, what do they do to non-membrane proteins? And now the numbers look very different. This is in fact not the accuracy, but the mistake. Okay? 60, I don't want to isolate the method, but you begin to see, so this is gram negative, gram positive, proteins without single peptide. Okay, some methods clearly do better. And we begin to see a big difference. And that ultimately is exactly the QOK for one membrane helix. That is a problem for methods. They also had a very high QOK, but then most of the proteins they see membrane helix is completely wrong, right? That's not measured in the one goes up kind of issue, and that is exactly the background probability. But this is proteins without single peptides. This is proteins with single peptides. So now we get into examples where some methods, and I do not even put single hydrophobicity based methods on here, but even some of the advanced methods get into very, very high error rates. In fact, let me just randomly put this one here. Is a quarter. Quarter means, so about a quarter of all the proteins are membrane helix, have membrane helices, 3/4 don't. So this is going to misclassify. In human, we believe we have 15,000 non-membrane proteins. This gets a quarter wrong, so this is immediately 3,500 or 3,700 misclassifications, a big number. And this 3,700 compared to the 5,000 membrane proteins. So almost this mistake actually means that almost every membrane protein is accompanied with one that is incorrectly predicted. Okay? So sometimes numbers, you have to think and translate numbers. In this particular case, the error rate translates to something in a membrane, in the whole proteome prediction that is slightly different. Very high, and we immediately see here that there's something to do. And okay, so am I going to go through 70 slides? The problem is I don't want to do another membrane prediction, or not on this one. Okay, I'm going to find a shortcut through the next slides, but on Thursday. I will call it a day for now and ask you for questions and see how I proceed from here. Thank you for your attention.